Alright, we're continuing our notes on genetics. We will finish talking about Punnett squares. Punnett squares, or a Punnett square, is a chart that shows all the possible combinations of alleles that can result from a genetic cross. And in our Punnett square, we represent that with an actual square that we divide into 4 quadrants. In that Punnett square, we write the male alleles on the top, usually. Sometimes it doesn't matter, but when we do this, for me, we'll put the males on the top. Males are symbolized with this symbol here, so I'll put that there. And then we put the females across the left side, which are represented with this symbol. And we put the alleles on them, 1 allele on each column and row, respectively. The boxes inside of our Punnett square represent the possible outcomes that the offspring can have, and they get filled in like a multiplication table. So, if we have our example with our P generation plants, both parents were homozygous for their trait. One parent, we'll say is the male parent, was homozygous dominant. It would have had 2 capital T's. So I put 1 T over the first column and the other T over the second column. Notice I do not put the T's. There's no more than 1 T per column. You cannot do that. And the other parent was homozygous recessive for short. So we put those alleles on the left side, 1 per each row. Cannot have more than 1 letter per each row. And this gets filled in like a multiplication table. When we do that, we have this T here. We're going to drop it down here. And we're going to take this T that's here and move it over here. We take this T here and drop it down here. Take this T and slide it over here. If I literally could drag these, I would, to help show you. Take this T and move it down here, and this T over here. We move this T down here, and this T over here. When we do that then, oops, I apologize for that. We have big T, little t in the first box, big T, little t in the second box, big T, little t in the third box, and big T, little t in the fourth box. And this is going to tell us some information about the F1 generation. So the alleles outside of my Punnett square were my P generation. And inside my box is the F1 generation. You should notice that those are exactly the same combinations that I showed you on our notes beforehand on the Ed puzzle. This is just a different way to show it. Now it's showing us probability. So let me explain the probability here. So how many, we're looking at what the possible offspring can be. And in this particular case, every single box is filled in the same. So we have a 100% chance that if we mate the homozygous dominant male with this homozygous recessive female, we're going to get 100% tall plants with the phenotype of big T, little t. I'm going to give you another example of a Punnett square. If I want to know about my F2 generation, I can do another Punnett square where I'm using these F1 offspring. So I'm using these right here. So I'm going to use one of them for my male parent, one for the female parent, and see what I get. I do remember that when we learned this before, we learned that Mendel's F2 generation got 3 tall and 1 short. And that's all we knew. So I'm going to show you more about that. Okay, so we've got, let me try to leave enough room for other notes here. Got our Punnett square with 4 boxes inside. Now we're going to do the F2 generation, which all were big T, little t. Big T, little t. So again, I'm going to slide this T down to this box. I'm also going to put it down in this box. Take this T, go down to this box and this box. This T is going to go over to this box and this box. And this T is going to go into this box and this box. It gets filled in like a multiplication chart. So here I've got big T and big T. And in the second one I have big T and little t. In the third one I have big T and little t. In the fourth one I have little t and little t. This shows us now how I get 3 tall and 1 short. So I've got a few things going on here. Let's look at phenotype versus genotype. See if I make a little chart. And then I'm going to put another for probability. Okay, so my phenotype, I have 3 tall. So tall is a phenotype. One of my tall came in this form of big T, big T. So I have a genotype of tall, which is big T, little t. But I also have tall where it's big T, little t. These 2 boxes here are big T, little t. So I have tall again, but with a different genotype. I just noticed I made a mistake up here. Okay, erase, erase. Trying to erase. Yeah. Okay, I'll just color over it. I apologize. We'll fix that. That's a big T, big T. Okay, so the probabilities. Now I look at how many big T's I have in my chart. So looking over here, I've got 1 square that is filled with big T, big T. And how many squares total do I have? 4. So I have 1 and 4. 1 out of 4. So what percent is that? 1 out of 4. Hopefully you come up with 25%. Then I look at my big T, little t. So I have big T, little t in these 2 boxes here. So that's 2 out of 4 boxes have big T, little t. What's the probability then? 50%. Okay, and then I'm going to look at my final box here. I haven't done anything with this. So this, little t, we have to determine the phenotype and genotype. So the phenotype is its physical appearance. We know that when we have big little t, these are going to be short. So my phenotype for that is short. And the genotype is the letter combination, little t. And we see that we have that in 1 out of 4 boxes. So 1 out of 4 again is 25%. So when we do this, all of your percentages should add up to 100 so that it's correct. Another way to, a couple other things to look at with this, when we look at, just as a review, this big T, that's considered homozygous dominant. Big T, little t is considered heterozygous, and little t is considered homozygous recessive. And then to point out another obvious thing, I guess, that I wasn't pointing out before, this chart here, we said, shows probability. So this is showing us not what definitely will happen in the future. It shows us what possibly could happen in the future. It says that there is a 25% likelihood that the offspring of this cross will be big T tall, homozygous tall. There's a 50% chance that the offspring will be heterozygous tall. And there is a 25% chance that the offspring will be short. So when Mendel did his cross, he did this a lot. He did this probably, I don't know how much is a lot for peat plants really, but every single time he did this, he took calculations, and when he calculated, when he added up and looked at the results, he found that when we add up this 25 and 50%, 75% were tall. Okay? And then 25% were short. He was able to then figure out these alleles by doing more crosses. So he could take these and cross them and see what they get. And he can look at the parents too. Okay, and here's another example with flower color. So this Punnett square has been done for you. In this case, we're looking at flower color. To me, the colors look purple and white, but they're using the letter B. So let's say they're blue and white. Well, they say violet, don't they? They say up here very specifically it's violet, but they use the letter B, so we'll say blue. So if we look at this, if I take 2 blue and cross them together and I get 3 blue to 1 white, then that must mean that blue is dominant. And so we use the capital B for blue, which is dominant. So we have actually 2 heterozygous parents. And we should have the same results that we had with our tall and short when we had 2 heterozygous that I just showed you, where we have 75% are blue and we have 25% that are white. Okay, when I look at the genotypes, we have 25% that are big B. We have 50% big B, and 25% big little b. So again, that's the probability of what their offspring could be. If we were to cross them and have 100 kids, it's likely that 75 of them are blue and 25 will be white. That's the likelihood. It does not promise those results. But the more you do it, the more close to those results you're going to have.